It is now. Hey,
There it
everybody,
goes.
it's Standing coming to you tonight with my guest, Kyle. He's got a degree in biology. He's been going at this for about 10 years now, so it should be an interesting discussion between us. We're going to keep it respectful. We're going to open it up just with a few minutes, up to 5 minutes of Kyle explaining his position without any interruptions. Then I'll do the same thing. And then we'll kind of just go back and forth in a conversation slash discussion format. I think our topic of today is going to be based on models, whether it's the biblical or evolutionary model, and see what better fits the evidence. So Kyle, you can go ahead, introduce yourself, and kind of take it off with your position.
Hey, I am Kyle. I have a degree in biology. I did most of my schooling at WSU, which is Washington State University. I focused on molecular biotechnology. I spent quite a bit of time in the lab there, genetically modifying zebrafish embryos with a heat shock transcription factor protein for Alzheimer's research. So my position is that evolution is the backbone of modern biology. The past 150 years since Darwin formulated his hypothesis, evolution has been substantiated time and time again, no matter how hard we've tried to actually actively disprove it. The evidence for evolution isn't just found in the fossil record, but it's found in genetics, and genetics is far more robust than fossils will ever be. Fossils tell us a little bit, but they don't really tell us all that much. And the downside of using just fossil evidence is that, well, we can look at the fossils and these creatures died, but when it comes to actually identifying them as whether or not they're the same species or separate species, it can be quite difficult. If we look at dog breeds, we can see Chihuahuas versus Great Danes. If we only found Chihuahuas and Great Danes in the fossil record, we would immediately classify them as separate species, just based on the morphology of the bone structures alone. And so when we look at the fossils for dinosaurs, for example, we don't know how much plasticity those species are experiencing. Like the dog example has a lot of plasticity within those breeds. So we have to resort to utilizing genetics, and it's more of an abstract thinking process than people are used to, where we have to analyze genetics and genetic patterns all through extant species, so currently existing species, and we can link them up based on patterns of inheritance found throughout the nested hierarchies. We can do this by analyzing things such as endogenous retroviral insertions. ERVs have a specific identifier denoting them as viruses. And we can find the insertion points, and we can find the patterns of inheritance where we would expect them to be if evolution was true compared to the rest of the genetics that is shared among closely related species. And we can formulate a pattern of inheritance, and this pattern indicates a branching tree-like structure. So this is part of the evidence that we use for denoting common ancestry. And we can also analyze how messy genes actually are. For example, we have a broken vitamin C gene that is fully functional in apes, but and other mammals, but we do have a broken vitamin C gene, and so we need vitamin C from other sources. And so we can't really biosynthesize it. More evidence is found in things like we have pseudogenes, we have part of our genes don't even have like any function that's a parent function. A lot of our genes are regulatory basis, and we're finding more and more function for a lot of genes that we thought that didn't used to have function, but it's still like a really big mess. And if we resort to intelligent design or creationism, then we wouldn't expect our DNA to operate in such a way that it looks ransacked. We would expect it to be a lot cleaner. We would expect everything to run a lot smoother. We would not expect so many genetic diseases to infiltrate populations so easily. Um, and uh, and so it looks as if everything did evolve. So that's pretty much my positioning for right now that I can think of.
Awesome. You know, I think you brought up some good points there. It was a good introduction. A lot we agree on as well, especially when it comes to fossils, right? Just like you said that the Chihuahua and a Great Dane, let's say they were extinct and we were to dig them up 1,000, 2,000 years ago, you know, a lot of the identification on those animals, on those, say, bones found in the dirt, would be based on, you know, assumptions, interpretations. So I do think that the debate is about genetics. I think it does come down to a genetic level. And I think with my, I guess, opening statement here, I would say, you know, the word evolution itself is a little bit of a slippery word. I think we have to define our terms, and I think we would agree that in regards to biological evolution, you know, it would mean, you know, a change in allele frequency in population over generations. So if we're talking about, I guess, change, you know, whether or not there's limits, I think in the biblical model we would definitely assert that there are limits. So if you by evolution mean, you know, dogs and, say, wolves are related through common ancestry, I don't have a problem with that. It's when you say dogs and, you know, whales or banana plants are related through common ancestry, that I think is where we're going to have the disagreement. But when it comes to our model, you know, we would say that species, they're not static, right? So neither are their genomes, obviously. They change over time. Sometimes, I mean, even randomly, and sometimes non-randomly, say like an epigenetic change or a mutational hotspot. Sometimes, you know, these changes are in pre-planned pathways, and sometimes even according to instructions, say from pre-existing algorithms. But, you know, according to our model, God obviously wasn't limited to creating static species. You know, so he placed a significant amount of design variation in each of his created kinds, which would indicate really that there could be a considerable amount of information stored, say, in the genome, whether it's, you know, in compressed hidden form. And then, you know, when this information is, say, decompressed, deciphered, revealed, maybe unscrambled, you know, this, whatever trait, whatever, you know, comes about from this cannot be used as evidence for, you know, pond scum to people evolution, since obviously this information was already present and stored in the genome. So we would say that Adam and Eve, they were given a significant amount of this design variation. So the built-in alternatives that God would have put into them, they're scrambled over time, and new traits could arise during this process. I'll get into, obviously, definition of kind here, but I think it comes down to, and I'll finish it off here, is, you know, and I'm sure we'll be talking about, you know, what the definition of information is. But I would say there has to be an observable mechanism that adds this new novel information to the genome, because technically, you know, the evolution of a microbe, say, into, I guess the best way I could put it, a complex organism, such as maybe a whale or a dog or a human, it really does require the addition of new genes, whether you know, we agree on, say, whatever this definition of information is. But, you know, if the simplest single-celled organism, and I think it's got about 500 protein coding genes, while humans, on the other hand, have over 20,000. So, if we began as microbes in, say, some primordial soup, as I believe evolutionary theory suggests, then obviously a lot of new genes had to be added by mutations. And obviously with the fine-tuning process of natural selection acting upon this information from mutation, that's really the only game in town for the evolutionists. So there'd have to be a lot of mutation adding such genes, not just altering or changing the existing ones. We're going to have to see, obviously, genes that make nerves and obviously all the enzymes that enable nerves operate, since, that is obviously absent from microbes. At the end of the day, they're going to have to be created if we evolve for them. So there are many gene families, I'm sure we can agree, in humans that are completely missing from microbes. So there has to be a viable mechanism for adding this genetic information if bacteria to biologists, evolution is even feasible. So I think it comes down to limits, and it also comes down to whether or not a genome can be built. We say God wrote the genetic script. Any change we see is based on that originally written genetic script. Maybe we can, I think that's all for now that I can think of to say, but I noticed you touched on a few things like endogenous retroviruses and non-functional DNA and things like that. I mean, maybe we can go into a point-by-point discussion on that, unless you have something else to say beforehand there, Kyle.
Yeah, there's just a couple of points that I want to bring up. So first of all, you pointed out the amount of protein coding genes in one organism versus, let's say, humans. Well, I want to say that protein coding genes aren't the only way to get phenotype. You can look at something similar to a pufferfish, and pufferfish actually have a lot of protein coding genes compared to all the other genes. While humans don't have very much protein coding genes compared to all the other genes. We have a lot more regulatory functions than pufferfish do. And so
so are you saying like humans would have a lot more non-protein coding genes, like these AREs, pseudogenes, junk DNA? Is that kind of what you're saying?
More so, well, somewhat, yeah, but more so I'm looking at regulatory functionality.
Sure.
Do you know what regulation is?
Yeah, you mean like regulating, like one gene regulating the, process of another gene, like the overlapping codes type thing?
Similar to that. So you can have one gene, for example, and then that gene can have multiple different functions and have multiple
Would that be like a heterozygotic gene, like multiple alleles per gene, multiple functions, regulation, expression, things like that?
Yeah, in a way, okay. Yeah. So, so protein coding genes aren't the only result of what makes a phenotype a phenotype.
Sure, I agree.
It's based on a lot more factors than that. Another thing I want to bring up is that you brought up that Adam and Eve had all the genes that are available in the human species, right?
I would say that God designed them with, say, tens of millions of DNA variety and DNA differences. And now it's just been a degeneration, a sorting out process since then. And that's how I would explain all the life we see around us.
So do you know how many alleles per loci is on the average? The average amount of alleles per loci?
I don't think I don't think you could say exactly how much, because it seems like we're seeing so many more roles and so many more overlapping codes. Like it looks like the DNA system is polyfunctional and constrained, because all that information is there, say, even in one nucleotide. It almost seems like there's a message forward and backward. But I mean, what would you say is the average? I mean, I'm not sure if they would have an exact answer to that. Is there an exact answer?
If memory serves, it's around 14.
Okay, but if it's like if it's a gene that's, say, identified as non-functional or junk, but now we're seeing that there is say, a regulatory role, or a role in gene expression in, say, that once defined junk DNA, would that prove that there's actually more allele frequency in that gene? Like, how sure are they of all these roles in genes?
You're looking at more of the expression. I'm looking more at just the alleles, not the expression of the alleles, but the alleles.
Okay, so like the varieties in each gene. So about 14 then.
Yeah. So do you know what a loci is?
Pardon?
Do you know what a loci is?
Is it like a site? Yeah.
A loci is a location.
Yeah.
Okay.
In the specific gene, right?
It's a location on a chromosome.
Okay. Yeah.
So when we look at alleles on loci, do you know why we have like 2 pairs?
Why we have 2 pairs on the chromosome? Well, because when my wife and I procreate, obviously, humans have 46 chromosomes, so my wife and myself are giving our zygotic information and alleles passed on to our child, right? So you're going to have those multiple functions per allele and per site on the chromosome, right?
Yeah. So you offer an allele, and your wife offers an allele, all right?
Correct. Yeah. Mm-hmm.
On the same loci, because sperm and egg each just have 1 set of chromosomes, and then when they combine, they create 2 sets, right?
Yeah, correct. You're right.
Okay. So if you have Adam and Eve, and that is one, and they procreate, that is 1 set from Eve, 1 set from Adam, correct?
Correct.
Okay. And the max amount on Eve is 2 that she can have, 2 alleles per loci, because she has a chromosomal pair per chromosome, right?
Sure, but I mean, wherever you're going with this, obviously, like I said, we don't know, because if we only speak 1 to 3% of the genetic language, we don't know exactly how much DNA variety, well, for one, is in us currently today, and especially what would be in Adam and Eve, right?
Well, what I'm saying is that if Adam and Eve had every single possible variation ever, then that would be impossible, because Eve could only have 2, Adam can only have 2, provided that they were completely different, genetically speaking, and provided that Eve was not a clone of Adam, which is what the Bible actually clues us in on, is that Eve was actually a clone of Adam.
Well, like, are you saying that if God would have given, say, Adam and Eve and all the original created kinds, say, these tens of millions of DNA differences, that would, provide the variety necessary for all the life we see?
I'm saying it's impossible. It's impossible to do that, to stuff all the genetic variation into just 2 individuals and then expect all this variation to come about from it.
Well, the thing is, if a lot of this information is hidden, say, in decompressed, hidden form, and then it's deciphered or unscrambled over time, because we're seeing now that a lot of our, and I think you're going to disagree with this, and you can tell us why, but it sounds like, based on the ENCODE project, I know that they're looking at, information in DNA that's transcribed, because 2% of the genome is actually protein coding, but we're seeing that a lot of these, what was called junk DNA or pseudogenes, AREs, they're actually, functional, just like some of the ERVs. So we're actually being mind blown at how much coding and DNA variety really is in our genome. So if we go back 6,000 thousand years to a point of least degeneration, because I believe in a process known as genetic entropy, I don't see, how that variety would be a problem. Go ahead.
Okay, so here's what I'm trying to explain to you. So I'm trying to basically get to the idea that you couldn't stuff all that genetic variation into a single species and then expect all this variation to come about from it. That's just not practical. There's something known as overexpression in genetics, and overexpression means that there's too many things being expressed. And so if you jammed all that information into organisms, then you'd have a problem of overexpression. There has to be a balance in molecular biology. There's a very good reason, for example, why.
But who's saying that all this information is being expressed? If it's being unscrambled or deciphered over time based on mutations and natural selection, whether randomly or non-randomly, why, and especially something like an epigenetic change that would be like a mutational hotspot in our genome, it's not all being expressed. But based on these I don't understand how that would be an issue if it's not all being expressed. A lot of it's in hidden form.
I'd like to give you an example.
Okay, go ahead.
If you put a lot of information into a computer, stuff a ton of information into a computer, what happens to the computer? It starts slowing down, right?
Yeah, it overloads.
Yeah, it overloads. Even if all your programs aren't running at the same time, it still overloads. And that's exactly what happens with human biology. If you have a system where a good example is, are you aware of what centromeres and telomeres are?
Yeah, so centromeres would be like the center of your chromosomes, and the telomeres would be the ends of it. And I think part of the reason why you age is because of the shortening or degenerating of the telomeres. I mean, that would come into play with the human chromosome too, right?
Exactly.
Yeah.
So what would happen, hypothetically, what would happen if we somehow were able to just extend telomeres? Let's say quadruple the size of the telomeres that we already have.
So if we quadrupled the size of the ends of our chromosomes in a hypothetical scenario, I'm not too sure what would happen.
Okay, so if we were to do that, it would end up clogging system. It would slow down the ability for our genes to be read, and it would slow down protein synthesis. And so if our telomeres are too long, then it's going to interfere with gene expression. And if our telomeres are too short, then it's going to be more subject to mutational changes. The telomeres act as a buffering zone. And so that's one of the reasons that we start getting problems when we age, is that our telomeres in becoming shorter, and then there's errors.
Is that a form of degeneration as these telomeres are getting shorter?
It is a form of degeneration.
Okay.
But this is why like humans cannot live forever, or practically no species can live forever. And I say practically because there are some mortal jellyfish out there.
And also as we age, like obviously our cells are mutating. Like after each cell division, how many mutations do we accumulate per cell division?
Are you talking about somatic cells or germ cells?
No, I guess reproductive cells.
Okay, so there's about 150, according to literature, I believe around 125 to 150 mutations per reproductive cycle.
Correct.
And evolution, we have to, when we look at evolution, we cannot look at somatic cell mutations. We have to look at germ cell mutations.
And that's because the germs, is it because the mutations in our germline, that's what is inherited in the next generation, right? Those are the mutations that are passed on, correct?
Correct.
Okay.
And mutations that occur in the germ cell also alter its own germ cells when they reproduce germ cells. Somatic cells don't do anything. They just infect the person that gets infected, and somatic cell mutations are like a leading cause of cancer, for example.
Yeah, I was just gonna. Yeah, that's a good point.
Yeah, or other diseases that might come about.
Yeah, that's why, like, if I were to get some type of cancer in one of those cells, and these cells start dividing, all intensely and negatively, I'm not going to pass that on to my children necessarily, right?
Not in somatic cells. In germ cells, there is a chance to have a predisposition to cancer, and that's because the way that cancer occurs is through these things called pseudo-oncogenes and tumor suppressor genes. And so you can have a predisposition if you have broken tumor suppressor genes, and there's a lot of locks in place before cancer begins, right? So let's say assume that there are 5 tumor suppressor genes, for example, and 4 of them are broken. They're already broken. So you already have a higher chance of getting cancer than someone that has 5 working tumor suppressor genes. Does that make sense?
Oh, yeah, that makes a lot of sense.
So also you have pseudo-oncogenes. Pseudo-oncogenes are kind of like the, if TSR genes are like the brakes, the pseudo-oncogenes are kind of like the gas pedal. And when pseudo-oncogenes end up mutating and become oncogenes, then it's like hitting on the gas pedal, right? Yeah. And it starts causing unregulated cellular growth, which we know as cancer. And so you can have a predisposition to gaining these diseases. But we also have to look at what evolution actually states when it comes to like various genetic diseases. If the genetic disease has a chance of harming the individual before or after reproductive success. And that's the kicker. That's what matters. Yeah. If a genetic disease, say, for example, a predisposition to Alzheimer's disease, or dementia, or something like that.
But I do believe a lot of those diseases can be environmentally related. Like I know a lot of heavy metal toxicity can lead to, Alzheimer's disease, dementia, things like this, like especially with all the toxins that we find, whether it's in the air, in the food or the water we drink. And I think that can result in a lot of these diseases as well. I'm not sure if you agree with that or not, but
G plus E equals P, right? You know what that means?
G plus E equals P? I'm not too sure. Yeah. Nope.
So genes plus environment equals phenotype. The phenotype is the physical manifestation of a gene.
I just want to ask you, and about like with natural selection, does natural selection act upon the genotype or the phenotype as a whole?
Phenotype.
Okay, so like if there's a mutation in say a single nucleotide, whether it's a point or inversion, insertion, frame shift, the natural selection is not going to just recognize that single mutation. It has to recognize the phenotype as a whole, right?
Yeah, because whether or not it's beneficial or detrimental depends on the environment. Correct. If you're in an environment, let's take oxygen as a really good example. Bacteria, anaerobic bacteria, are widely known to just be destroyed by oxygen. Oxygen is actually toxic. Not many people know this from what I've seen, but oxygen is actually toxic because of superoxides, and the superoxides actually end up harming us. We're basically on fire, in a way. Yeah. Because of these superoxides. But we have a enzyme that is able to take care of these superoxides. And this enzyme evolved very, very long ago. In some bacteria that went from anaerobic to aerobic.
Is this the E. coli Lenski experiment? Because I know that was a change that they did observe in the bacteria, right?
I'm not sure on this particular change. I don't know too much about his experiments, except that I know that he caused E. coli to start digesting citrus. And that is very unusual for E. coli to do.
I think it was aerobic citrate. But I mean, if you look at it, it involved the loss of digestion. I mean, it involved the loss of control of, I think, the normal anaerobic citrate digestion. But I thought that that's where you're going with it. But that's fine.
Well,
Keep talking about
so
what you're.
what I'm saying though is that this mutation ended up evolving to be able to handle oxygen, and so from this,
Actually,
this
is this something you can demonstrate, or something that is theorized or hypothesized? Like, how long ago was this that this
oh,
change
we
happened?
can find the gene in aerobic bacteria.
Is this like a non-functional gene, or is it a?
No, it's a completely functional gene that creates an enzyme that is able to take care of oxygen.
But I mean,
It's
how do you
able to
know that?
metabolize it.
Yeah, but how do you know that gene evolved this ability? Did you observe this? Like, I'm not
Well,
sure. Like, is this a story, or is this like something that has been observed? I just want you to clarify.
define what you mean by observed.
Okay, so you
Because we
see
can.
this bacteria that has this gene that has this function and trait regarding the aerobic or anaerobic or whatever. Like, is this something that you see this bacteria possesses in its genetic information, or is this something that you're theorizing evolved? Like, are you trying to explain how this certain gene evolved?
So,
That ability?
what I'm trying to explain is simply the model of what these researchers have expressed, and once we understand the exact model of what they're saying, then we can go back and then test the model for accuracy, right?
Sure. Yeah.
So currently, I'm just explaining the model of what these experts are saying.
But is this a story that they've concocted on how this specific gene evolved the ability to do this important function? Like, or like, is there?
Well,
Uh,
I don't like the word story because that brings with it baggage.
well, because if we're looking at this bacteria and the gene and the function that it has, we see that it has that function. But it sounds like the story part comes into play. When you say, "Hey, this is how this certain gene evolved this ability." I'm not really sure of the exact situation that you're talking about. So that's why I'm just trying to clarify. Like, is this something that they've observed in the lab, or something that they're hypothesizing has happened in the past? That's all I'm
So,
saying. I'm not trying to
okay,
be
well,
argumentative.
I like the term hypothesize. So this is a law of hypothesizing. Like I said,
Okay, I understand.
understanding evolution is very abstract thinking.
Yeah.
It's not as direct as creationists want it to be.
Well, like, what do you mean by, because like I said, I mean, the word, obviously, evolution can be, because do we agree that the word evolution means, especially biologically, like a change in allele frequency in populations over generations? Is that an accurate representation of what you mean by that?
So in terms of evolution, yeah, that is essentially the biological definition of evolution. And I guess we should really clarify what I mean by evolution. That's one of the
Yeah,
things I
yeah.
meant to bring up. Um, so evolution can mean a lot of things. Like it depends on the context. Like for example, if I'm a forensic scientist or like a criminal psychologist, I could say that the killer is evolving. All right, that doesn't mean that the killer is becoming a higher life form, like Pokemon, right?
Yeah.
That just means that they're changing their strategy. If or they're becoming worse or something like that. It's not my field of expertise. I just
Okay,
heard
well,
that
I
phrase
just
from
don't
my
want
mouth.
to straw man your position. No problem at all. It sounds like we agree, at least on the biological meaning of evolution. I don't want to straw man your position. And you've probably heard this question a million times. And the only reason I keep asking it is because literally everybody I ask gives me a different answer to it. So I just want to see, what you believe about it, and whether you believe there's, physical limits, genetic limits. But like, do you believe we can say get a dog as small as a flea? Because we do know there's insects as small as a flea, like a flea. Or do you believe we can get a dog as big as an elephant? Because we do know there's animals as big as an elephant. What's your answer to that?
Okay, so this is a little bit tricky to understand
Sure.
because there are limits, but there aren't
What do you mean
the
by that?
limits that you think. And so it gets a little bit complex, and it deals a lot with life history.
Sure.
So it depends on the previous mutations, and previous mutations pave the way for future mutations and
Yeah.
future adaptations. So a good example is, let's say, the organism managed to start metabolizing oxygen, and now it can live closer to um, the atmosphere, right?
Is this the bacteria that you're talking
Yeah,
about with
yeah.
the
Now it can live
gains?
closer. So I'm going to go over, and it may sound like a little bit of like a story at the moment, but just go with me here of what I'm trying to explain. Okay,
Yeah,
so
okay, yeah, go ahead.
so it can live closer to the atmosphere at the moment, and since it now is able to live closer to the atmosphere, then it could evolve other things by utilizing the oxygen that it now managed to figure out and utilize, and it creates a scaffolding structure, and it builds up from this. For example, you will never be able to crawl on land without having this ability to take care of the oxygen in the atmosphere, right?
Yeah.
You'll never be able to do that. So developing oxygen was a key component to higher evolving.
But is this
And
something? Am I just going along with the story so far? Is this
yeah, yeah,
something
yeah.
demonstrated in the lab? Okay, keep going.
So I'm just explaining.
Are you there, Kyle? Um, Kyle, are you still there? I think your microphone might have went out. Just one second. Kyle, can you try fixing your computer or something? We can't hear. We can't hear your. We can't hear you at all right now. Looks like Kyle's microphone may have gone out there, but I'm gonna send him because we seem to be having a good discussion right now. A lot of the things that I was trying to move towards, especially when it comes to the mutations that build up in our germline here, especially if it's neutral mutations, because the genetic entropy and the degeneration process, it occurs at a personal level with us as we age and as we die, but it also occurs in a population level, right, with the accumulation of these near neutral, very, very much deleterious mutations. So that's kind of where I was trying to get Kyle to go with this, and obviously pond scum to people evolutionary theory has a lot of fancy stories to explain certain things, and obviously if it's not observed or demonstrated, but it's a story concocted by people with large government grants. I mean, it's not exactly empirical science here, but we're just going to take a quick break here, and I'm going to see if he's coming back in. I'm going to send him the link because we are having a good conversation here, and I don't want him to. There we go. I just sent him another link. We'll see if he's able to make his way back in because the broadcast is still going. We'll give them a few more minutes here. Not sure what the technical difficulty is exactly, but so we'll give them a few more minutes here. But especially when it comes to the junk DNA, the non-functional DNA that he was talking about, we do know that there are a lot of functions to this, especially the fact that we only speak 1 to 3% of the genetic language. It's almost like putting, a kid in front of a car, the hood of a car, and saying, hey, explain to me what all this, mechanical baggage does, and then remove anything that you believe has no purpose or no function. And, that kid's going to start tearing things out left and right. But that's based on ignorance. So, we only speak 1 to 3% of the DNA code, but we're finding that a lot of these non-protein coding regions, they got a ton of functions, whether it's regulatory, they even do things like control telomere length, that, kind of helps and serves as a molecular clock to control aging. They even, help in embryological development because, especially, in the embryo and a baby, they have a huge developmental process and a lot of processes going on before they become, a fully grown baby. And a lot of those non-protein coding regions, they serve as, regulatory aids in the embryo. And it's pretty amazing because at the end of the day, a lot of it's almost like a construction site. You got engineers, mechanics, workers, tools, and a lot of work is being put into the building of, a building. And at the end of the day, when that building's built, all that stuff is now not needed. And we see that in our genome as well. I mean, the biblical model would predict limits. It would predict changes that can be safely categorized in a loss of information, loss of function. We predict, obviously, this fully functional genome with some obviously mistakes based on genetic entropy, because these accumulation of near neutral mutations over time, they're degenerating and degrading us as a population. And the arguments that I've seen against genetic entropy here, especially beneficial mutations, they're so rare, and these evolutionists, I don't think they can grasp just how difficult it really is to build a genome apart from design, because a population, it can be undergoing genetic decline due to these vast numbers of slightly deleterious mutations, even while selection can be amplifying a handful of beneficial mutations. So these beneficial mutations are rare. And even if you get one and you can demonstrate it sufficiently, it's just noise compared to all the very, very many deleterious mutations that we know are building up into our genome. I mean, a species, and I think I said this to Jackson in my last debate, it can undergo minor adaptive fine-tuning, obviously, to its environment based on that genetic algorithm God placed in our genomes. But, we're still degenerating in many other ways, just like a car. I'd say a 10-year-old car, it's degenerating in all possible ways. And you can install new windshield wipers, you can replace the tires, replace the brakes, but has the car stopped degenerating? Just
I'm
like our.
back.
Oh, hey, brother. We kind of went quiet for a couple minutes, and I thought it might make for a boring hangout, so I kind of just went on a rant there. How you doing, brother?
Good, good. I'm currently on my phone. It's
I
plugged
figured there
in.
was a technical difficulty, so I kind of just let you. Was it your microphone, or like was it my computer, or do you know?
No, my roommates ended up bumping my internet and completely kicked me off, and I'm having trouble even signing back in now. So, it's something I'll have to take care of. I don't have very much battery life, so I'm hoping this doesn't take up um, much battery to do the hangouts.
I guess
Um,
I guess since what I was talking, because I know we were talking about how the telomeres shorten, how we age based on these accumulation of mutations. But I'm curious of your opinion on the work of Dr. Sanford. I know obviously you probably think it's a fraud, but I just want to know your opinion on it, because if we have this accumulation of, say, these near neutral, very much deleterious mutations, if you can see that entropy process is why we die. How come you can't see that, as a population, these accumulating near neutral mutations are not going to degenerate us to the point of extinction? Like, how do you feel about that information there, Kyle?
well, there's only two words that really come to mind, and that he's wrong. Uh,
Why? But the
well,
thing is, in a lot of my debates, I've never heard a good argument as to why. I'm wondering if you have a good argument.
Okay, so we have to really understand what is going on with why we die, and that's mainly based on somatic cell mutation buildup,
Yeah.
and evolution occurs on germ cells, not somatic cells, and so germ cells are able to get mutations that are functional and can pass on to further progeny. And there are a lot of checks and balances that actually go on uh, to germ cell mutations. First, they have to pass the first test, which is be able to create an offspring in the first place. Some mutations don't pan out, and there can't be any organism that comes about through reproduction. It will get destroyed by the body's own processes. This is where miscarriages come in, right? And a lot of women end up getting miscarriages, and a lot of times it's due to detrimental mutations. And so after that, you then have to go through checks and balance of whether or not the mutation is going to assist the organism with being able to survive. Um,
Would you say we're all mutants, though? I mean, based on these accumulation of mutations over time through our germ cell, like, are we all just mutant? What, and some more mutant than the other?
we're all completely mutate. We're all mutants, but we're not like more mutated than any other person.
So, like,
Um,
if natural selection is going to act on the phenotype, like we talked about earlier, is it natural selecting just deciding and distinguishing between one person who's mutant versus another person who's mutant?
uh, Sort of. So all life is based on in competition. Some alleles are better fit to survive than others, and the better
Yeah.
fit alleles have a better chance of being passed on. And that's what really matters in evolution, is what are the chances that an a individual mutation, um, given the package that's found in, is able to pass on to another organism. And this is where a lot of Dawkins' work in the selfish gene comes in. And there are
Okay.
some biologists that don't like the idea of the selfish gene, but there are some that do. And so there's a little bit of contention in that department. But in terms of and this is what we mean by like scientists are in contention on a lot of issues dealing with biology, is that they are in contention on the details of what's actually going on. And this is one of the details that Dawkins proposed was the selfish gene. Um, so
Well,
we look at.
actually, sorry, I heard an echo there. Do you have? You just got to make sure that there's no. No, it looks like we're good now. Okay, keep going, Kyle.
Okay.
It only echoed there for a second. Usually that indicates there's another YouTube video on in the background, but whatever it was, we're good.
There's no YouTube videos on in the background.
Like if natural, okay, so like you were saying with obviously survival of the fittest and these alleles that natural selection chooses in these traits, like would it be safe to say, like, if we put all the dogs in the world today, say we put them into Alaska, natural selection would act upon that allele, say for thick long hair, and then over time we'd see just a population of, say, huskies. But say you took that population of huskies and put them into the desert of Australia, wouldn't natural selection then act upon those existing alleles, and eventually you're just going to have a population of, say, dingoes, short skinny legs, more adapted to the desert? Like, would that be a valid explanation on the fitness?
Sort of, but so one of the misconceptions that I fear you might be unintentionally going at is that putting like a Chihuahua or a group of Chihuahuas um, in Alaska won't, if you reproduce them all the time, they won't.
Well, I think those Chihuahuas would probably just all die out.
well, if provided they survive, I'm talking about. Eventually, you won't start getting huskies because huskies already exist. Does that make sense?
Exactly. Yeah. No, I agree with that. So obviously all your Chihuahuas, all your poodles, because you can't take two Chihuahuas or two poodles and artificially breed them back into, say, a wolf. Okay, because that speciation has gone too far based on that sorting out of the genetic information. That's why when people use examples about speciation, I'm thinking it's the opposite of what Ponce come to people evolution needs. But if you were to take two wolves and artificially breed them, over time you could get a poodle, you could get a Great Dane, and you could probably get a Chihuahua, right? Because then you're working the other way. What do you think about that?
No, because Chihuahuas, Great Danes, etc., already exist. If you take a wolf pair today, you would not be able to artificially select them and then recreate a Chihuahua eventually sometime down the road.
You don't think you could?
No, you might get a breed that might resemble a little bit like a Chihuahua, but it wouldn't be Chihuahua.
Okay, could you get two Chihuahuas and over time breed them into something that looks a little bit like a wolf?
It's possible that, yeah, you could do that, but it wouldn't be.
I mean, I don't think that they have enough allele variation and enough heterozygotic change to bring those poodles or Chihuahuas back into something like I think that information has been sorted out far too much. But I guess we're just theorizing here. But I don't think there's enough variation in the genes of the gene pool of a couple Chihuahuas to get into something like a wolf. But I don't know. Maybe we're just playing semantics here.
That's where mutations come in.
What's that?
That's where mutations come in.
But like, if these mutations are overwhelmingly deleterious, and we don't really see too many of these mutation adding, information adding mutations, I don't understand how you're going to be able to go back to something like a wolf. Can you explain how that would be possible? Acting on the existing information and all.
Well, first of all, we have to find what information even is.
Sure. I mean, I would probably. Have you ever read the book? In the beginning was information by Dr. Werner Gitt.
To be honest, I have not, but I don't take Gitt very seriously at all.
Well, I'm just saying, like, a definition of biological information, I don't think it's easy to come by. But if I had to give it, maybe a technical definition, like, I guess I would say it's more of an encoded, symbolically represented message, say, conveying expected action and intended purpose. And I like the way that Gitt presents information. He presents it in like a hierarchical level, 5 of them. Okay, so statistics, syntax, semantics, pragmatics, and I think abobetics or something. So the way that he defines it, I think, is probably the best way to define it. Because I know there's also Shannon information, but I think that's an improper use in regards to biological information. Go ahead there, Kyle.
So, in terms of biological information, what I like to use is more so analogies. Nucleotides are very similar to zeros and ones on a computer, right?
Yeah.
And when we look at information on a computer, we analyze these as bytes, right?
Yeah.
So I usually, when I look at information, I look at, well, is the mutation adding additional nucleotides? If
Yeah.
it
Yeah.
is, then it's increasing information.
Well,
And
increasing the genomic size, but without having any actual meaningful information, wouldn't be adding anything novel. So I think that's where we would disagree there. Go ahead.
so it might not seem meaningful to you, but it can most certainly be meaningful to the organism.
Well, if obviously life was, say organisms, obviously, for life to exist, I'd like to see a hierarchy of, um, you know, comprehensively regulated, information driven metabolic functions. I mean, there's just so much to life that, can mutations and natural selection build up the genome to what we see today? Go ahead, Kyle.
Yeah. So one of the misconceptions that you're bringing up constantly is from Sanford, and he says that the organism itself is in a constant spiral of decay,
Yeah.
essentially. And so, if we take Sanford's work as being true, then we can easily test it. Uh, just let's look at something that has a very small genome
Like a
and
bacteria?
What was that?
Like a bacteria?
I'm thinking more virus.
Okay, maybe like an RNA virus or something like that.
Yeah, like HIV.
Sure, yeah, that's a good example.
Okay, so very, very small genome, all
Yeah.
right, reproduces a lot very, very quickly.
Is there a high mutation rate though?
There is.
Okay, yeah. Are there short generation times?
What do you mean?
Obviously, right? In these viruses, are there like it is a lower genome versus a higher genome, like humans, right?
Yeah, yeah. So
Okay,
I don't like
okay, go ahead.
using the term higher or lower genome because that doesn't really make sense. There's just different genome compositions.
Sure.
So basically, what I'm saying is that if Sanford is correct, logically, we would expect HIV to easily just wipe itself out.
Yeah.
But it doesn't. It sustains in the population. It changes. It has a high mutation rate. It evolves very, very, very quickly. And it changes. And it is able to bypass our genome. It can frame shift itself and still remain functional as a defensive mechanism. That's fascinating, right?
But
So,
if but in regards to the viruses, and I guess even bacteria, I understand that it is evident that they can persist in spite of the genetic entropy, like you're saying. But can't these viruses and even bacteria, can't they be preserved in a dormant state for even thousands of years? Like, isn't that a known attribute of viruses and bacteria?
if you put viruses in ice or bacteria in ice
Yeah,
and
they'll be
freeze them,
preserved, right?
they can be preserved. Which is
So
actually one of the a little bit tangent here, but that's actually one of the concerns about global warming is that if we melt the ice caps, where it's possible that we could release a virus um, that life is not accustomed to, or a bacteria that life is not accustomed to,
Yeah.
and hasn't evolved with to be able to build up those resistances against, and it can be very, very detrimental to a lot of species.
So yeah, no, and I'm sure that some pretty intense viruses have been released, throughout history. But like, even if, like, say the most active strains, say they just continuously died out, I don't know, after 1,000 years or so, new strains could really be continuously receded into the environment, like you're saying, say from natural dormant reservoirs. So I mean, there's obviously a lot of reasons why something like an RNA virus, HIV, bacteria, they're still around, even in spite of genetic entropy. Because I've read a rebuttal. I think it was a blog called Letters to Creationists, and he used that argument, and it didn't seem very convincing, because obviously the argument of genetic entropy, we're applying that to higher genomes. I know you don't like that word, but we're applying it to mammals such as humans. Go ahead.
So, a couple things. One, we're just using the idea of a small genome to test what you're saying because it's just a model organism to be able to test that situation. If DNA just degenerates constantly, then we would expect it to degenerate at a very, very fast rate right in front of our eyes with something that reproduces quite often with such a small genome. And we just don't see what Sanford is saying in bacteria or in viruses. But if you want to look at higher organisms, I don't like using humans as a good example of that. And that's primarily because we use intelligence and we have made modifications to the environment to better suit our needs. So we don't need to change to suit the environment's needs any longer. We
Well,
have
like,
medical facilities. And because we have medical facilities, we can save people that would have died and not been able to pass on their genes.
but
And
our
that's
no,
creating artificial buildup of negative traits.
but like, wouldn't beneficial mutations have to be not as rare as we know and see them to be, in order to, say, combat this accumulation of deleterious mutations in our genome? Like, how rare are beneficial mutations, do you think?
Rarity doesn't matter, honestly. A lot of people think that rarity does matter. A lot of people that are unfamiliar with the science of evolution and genetics always look at the rarity of beneficial mutations compared to detrimental mutations. By the way, neutral mutations actually occur at a higher frequency than both. Uh, and I will admit, detrimental mutations do occur at a higher rate than beneficial mutations. But we
Yeah.
also have to look at
And
a lot of factors. One of them is the environment. If the environment is harsh and it sets the bar very high for who is able to reproduce, then it's going to keep a lot more beneficial mutations in the next generation and get
what
rid
would be
of.
I just think, what would be an example of a beneficial mutation? Just so we can maybe get specific, because you're saying obviously a beneficial mutation, whether it's rare or not, it's obviously necessary. We agree that. Like, what would be a beneficial mutation that natural selection preserves and fixates into the genome?
A good example is something like CCR5 delta 32.
And what's that exact? Which one's that exactly?
So HIV is capable of attaching to a certain location on the cell receptors, and it modifies the cell receptor, preventing HIV from being able to infect. And so, in an environment that is heavily influenced by HIV, the CCR5 delta 32 mutation will be able to protect against it and proliferate more CCR5 delta 32 variants,
Is
while others will get sicker and die off a lot younger.
Is this mutation? Is this more of like? Is this based on a heterozygotic advantage, kind of like sickle cell anemia, or is it something that has no deleterious effect? Because we're looking at net loss versus net gain, right?
So sickle cell is based on incomplete dominance. What I mean by that is that you can have one gene and it would be like the sickle cell gene, and then another gene that is completely normal, and then you'd have like it looks to be like a more of a hybrid between the two, um, and so you don't end up getting sickle cell, but you also maintain the benefits of having that single allele, right? The CCR532 is different.
Well, because I'm just trying to because I know with sickle cell anemia, it confers a resistance obviously to the malaria parasite, but as a result, it's producing deformed hemoglobin molecules. At the end of the day, it's still deleterious. So, are you saying this one has no ultimate deleterious effects? Like if I looked into it, I wouldn't find any literature that gives us a very degraded advantage.
Well, here's the things about mutations is that a lot of times they're beneficial in one area, but then detrimental in another.
Exactly. Yeah. So that's no help to evolution.
But it is because it depends on the environment. A good example, let's take sickle cell, for example. Sickle cell is a good example of being very contextual. In America, sickle cell would be strictly detrimental. But in Africa, where malaria is present, sickle cell gene is very beneficial. And the reason is because let's say, and I'm just pulling numbers out here to give you a conceptual example, because I don't have the numbers in front of me currently. But let's say 25% of the population ends up dying from, and then 25% end up dying from malaria.
Yeah.
And the 50% actually survive to reproduce, right?
Sure.
So if we look at this, then we can also see that let's assume that sickle cell never entered into the population at all.
Yeah.
And let's say only 20% of the population survives and 80% die of malaria. Okay.
Well, but and keep going with this. But like this mutation is just to reiterate or understand, it is dependent quality based on the environment, though, right? Because you're saying it has deleterious effects in one environment, but it has a beneficial outcome in, say, another environment. Am I correct to reiterate that?
Yeah, a lot of mutations, I would say most mutations that I can think of depend on the environment. It's very contextual basis. Some mutations are going to be beneficial in one environment, but detrimental in a different environment. For example, flippers wouldn't do us any good on land, but they certainly benefit dolphins in water.
Well, yeah, and dolphins have flippers. That's obviously environment dependent as well. And obviously we have no mechanism that can take an animal, that has no information present for, say, something like flippers to gain the novel information that would lead to a new body plans and structures such as flippers. Because if you're saying that these types of examples, like for example, the HIV one, these people, they have a mutation, you're saying, okay, that prevents HIV. Is it preventing it from entering the white blood cells and then therefore replicating it so they're not going to develop AIDS? Is that kind of the
Essentially.
Okay, so now these
and
people.
It also has another effect. Literature that I've read say that it became more in prominence after the Black Plague swept through Europe. It was only in, let's say, I think it was like 1% of the population, and then it became in 10% of the population, or something like that. And that's what some researchers are theorizing, is that it protected against the Black Plague.
Well, from the material that I've read is that the individuals that say get this beneficial outcome, they're going to be at a higher risk of developing illnesses associated with like West Nile virus and hepatitis C, right, which is also a virus. So obviously, if we look at that, it's still a mutation only beneficial in a given environment, such as if a person was exposed to HIV. But it is possible that the mutations would not be beneficial. And if you don't agree with me on this, let me know. In other environments, such as if the person were exposed to West Nile virus or hepatitis C. So once again, the benefit of any given mutation, and especially this one, it's dependent quality based on the environment. So is this the type of change and mutation that you believe is going to take your bacteria to biologist? I mean, is this your proof for evolution?
Everything is give and take. Let's say
it's still deleterious. I don't understand how that's evidence. I don't understand that that's the type of change that's going to turn your fish to fisherman. I mean, enlighten me, but I don't see it.
Okay, so you want to discuss, let's say, fish to fisherman, right?
Yeah.
Let's look at dolphins and let's look at bats, and try to understand a concept between the two. Both are mammals, correct?
Correct.
And both are unique in that they are encompassing an environment not normally associated with mammals, correct?
Correct.
So bats fly and dolphins swim. But there is something similar in both of their phenotypes. Bats have wings, dolphins have flippers.
So are you talking about homologous structures like the is it the radius that's the same in both, like the arm bone type thing?
Sort of. So what I'm looking at primarily is the evolution of webbing, and understanding the concept behind webbing. And looking at bats, the way that they got their wings is based on it's all based on the advancement of webbing, and we've observed webbing occur in humans, for example. Toads webbed.
And you can demonstrate this, or is this like another story? Just to clarify.
We can demonstrate this. Toes can become webbed. Human toes can be a baby can be born with webbing.
Well, isn't it that skin, that excess skin, from the pre-existing information is now emphasizing, like, obviously, I would say that webbing is a negative, feature or trait. But is that the same as the webbing that you're referring to in, say, a bat wing?
Well, it depends, right? Like, it's very similar concept. The webbing in a bat wing, or the webbing on a flying squirrel, the excess skin actually does confer an advantage, and that advantage is being able to glide or fly by creating surface area, and being able to catch that wind.
It's a positive advantage. I agree.
What was that?
I agree. That's a nice advantage and a nice trait to have.
Yeah. So in terms of the development of wings, they used to be arms, and you'll never find a organism except for insects that. So I'm more so looking at like birds or mammals that their arms don't become wings. Like, for instance, we would never find a dragon.
We would never find a what?
A dragon. And I don't know too much about insects or how they developed wings, but I do know that the way that birds developed wings is based on their arms, because birds developed from a lizard-like reptile species, and the scales turned into feathers, and over time, of course, and the arms themselves began to get a webbing, like a bat developed a webbing, and from this webbing, uh,
Are
they
you saying
started
you've seen?
using.
Are you saying you've seen these bats go from no webbing to webbing? Is that what you're saying?
I'm saying that we can look at the genetic evidence and the phylogenetics and the homology of structural
Well,
concepts within birds and bats, and we can truly start understanding how these mutations started occurring and the
well, like,
patterns that we see.
okay, but isn't it true that like with these homologous structures, aren't they based on a genomic level? Like if you're looking at the phenotype and you're looking at this homologous structure, aren't they based and coded by totally different genes? I mean, how does that make sense? That seems more consistent with design than with universal common ancestry. And when it comes to phylogenetic systematics, at the end of the day, I feel like that's based more on an assumption that, say, bacterial biologist evolution is true. But if genetic entropy is true and a loss of information regarding to mutations is true, these phylogenies are more so just the result of a false assumption. Go ahead.
So, are you referring to the fact that two different genes can result in the same or a very very similar phenotype?
Like on these homologous structures, say whether it's um, the humerus on different mammals, because you're saying that those are homologous, therefore they descended from a common ancestor. But those structures are actually at a genomic level. They're coded on totally different genes. So I'm just trying to understand how that's possible. Is the explanation is that would be like a more of a convergent evolution, or like what's the explanation for that?
A lot of times it can be convergent evolution. Yeah.
But isn't that more of like a rescue device? Like, hey, we can't explain this, so convergent evolution, so that structure evolved differently instead of them diverging from a common ancestor. It just happened. Like, for example, like dragonflies, bats, birds. Their wings would have evolved in a convergent manner, right?
Close the door, please. What's that? Close the door, please. Sorry, what was that?
Like for example, dragonflies and their wings, and say like a bat's wings or a bird's wings, wouldn't that be an example of convergent evolution because they evolved those wings separately?
Yeah, or you can look at dolphins and fish,
Yeah,
and the reason
that's a
the
good
reason
one.
that the reason that we say that this is not just a rescuing device is that if they evolve in the same, it makes sense, right? If they evolve in the same or similar environments, then they're going to get the same or similar mutational aspects to them, and if but we also have to look at their life history. They aren't going to be exactly the same. They're going to be somewhat different. They're going to have traits that are associated with their ancestral heritage.
But if,
We look at whales and dolphins, and we can
but can you look
identify.
at the history of these, or like, are you saying you can look at the evolutionary history of something like a dragonfly? Like, what did a dragonfly evolve from that? If you can look at the history, where's the transition from, say, a single-celled organism up to, obviously indirectly, up to a dragonfly? Where's the history of that one?
I'm not an entomologist, so I don't know too much about insects, unfortunately.
But what
Um,
about the history of? Okay, what about the history of, let's say, a bat? What's the common ancestor of a bat? What did that evolve from?
okay, so with bats, chances are it's going to be like more of a rodent type
Okay,
common ancestor.
so it's a
That
story. So we don't know the history of
well,
the evolution of these animals, because you're just speaking it like, hey, we know the history of it, as if you can watch it on a movie or reading in a book. It's all stories
like I said, a lot of this is
concocted
conceptual.
with a government grant.
Well, like I said before, a lot of this is conceptual. You really have to understand to think conceptually, and be able to understand that this is all a lot of this is looking at this from a conceptual standpoint and being able to analyze the evidence after we understand what the model is saying. So once
Well,
we understand what the model is saying, then we can go into the evidence and say, well, does the evidence support the model?
okay, how come it's not logical? Because you're saying it's logical to conclude universal common ancestry based on these homologous structures and also based on homology, you know, at a genomic level. But if I were to take, say, Microsoft PowerPoint, Microsoft Excel, and Microsoft Word, and I were to sequence all the lines of code in each program, I'm probably going to conclude that there's 95% similarity between the programs. But at the end of the day, that's not going to prove that they, you know, evolved from Morse code billions of years ago. I mean, that proves common design. So if there's a competing explanation and a competing model, how come we're not acknowledging that? You know, is common ancestry the only explanation for the homology we see?
It is.
Oh,
We can't.
okay.
Yeah, yeah. And let me explain. We can't really use things that we know are made by humans and then
Why
say,
not?
well, this is intelligent design, so life must automatically be intelligently designed. That's a little bit of an appeal to complexity
Well,
right
if
there.
we see lines of code in Microsoft programs such as Excel, PowerPoint, and Word, and we see lines of code in the genetic information system of living things, that is a valid conclusion. I mean, obviously you disagree with it, but these similarities do prove common design. It is a competing explanation, whether you say it's not. I don't
So
understand your thinking on that.
here's the reason that I do not accept the idea of a common design is that if we evoke common design,
Yeah.
then that would mean that we would also have to ask the question of what designed the designer.
Okay.
It's
Sure.
infinite
Yeah.
regress solution.
Well,
And
obviously we would trace it back. You know, obviously that would be a separate who's the designer. You know what I mean? Like we can conclude if we're walking through a forest and we see a painting hanging on the tree and I see nobody around and no footprints, I'm going to say, you know, did this painting come about through natural evolutionary processes or, you know, was this intelligently designed? So if I conclude it's designed, it doesn't mean that I know the designer. I may never know the designer, but it doesn't mean that we can't use that as a competing explanation. Go ahead, Kyle.
but the problem is that we have to ultimately ask after that, well, who designed the designer? And eventually you're going to reach to the point where you say that the designer doesn't need a designer, and then that's just special pleading.
Well,
You're
it's
you're
not special
saying
pleading
that
because according to Christian theology, you know, God is the uncaused first cause. So at the end of the day,
that
that's
is special
a
pleading.
theological based question. Obviously, if you're going to go back to who designed the designer, but just like in our email when I said, you know, I use the second law of thermodynamics, which is the law of increasing entropy. You know, the universe is winding down. You wind it back up. Who wound it up? You know, that points to a point of least entropy, a point of creation. So I mean, there's just so much evidence that, you know, there was a creation, and you know, who's the creator? Sure, we can talk about that, but common design does seem like a pretty logical and valid conclusion based on the evidence. Go ahead, Kyle.
So it's still a special pleading argument because then you have to ask yourself what created the designer, and if
Well, I
we
don't as a
put it
Christian
down,
because
well,
you believe
if we
he's
boil it
eternal.
down, the simplest explanation with the least amount of assumptions, Occam's razor, is that God doesn't exist and the system itself is um, is self-creating and self-organizing and self-destroying.
Okay, so
Um,
you believe that you know, princes and frogs sharing a common ancestor. You think that's the most simple explanation? Because that sounds like a fairy tale to me, to be honest with you. It doesn't sound simple at all. Pine trees and elephants are
evolution,
related. That's a simple explanation.
evolution and common ancestry is the most simplest explanation with the least amount of assumptions, because we have the evidence to support it. Quite frankly,
Well, I haven't seen any evidence yet. I mean, I seen you bring up ERVs, okay? And at the end of the day, now we're even seeing that these endogenous retroviruses, you know, functional ERVs would depend heavily on their position and their sequence. So If they were positional and positioned slightly wrong, or they even had the wrong sequence, they wouldn't be functional. And yet, what do we see? We see that they're actually functional. You know, they aid in transcription in a fifth of the genome, and that's actually strong evidence that, you know, these endogenous retroviruses are not actually the product of retroviruses. So even that, as a big major proof, is now falling apart, just like all the rest of the junk DNA. Go ahead, Kyle.
That's incorrect.
Why is that incorrect? We're seeing that there is function, and that they are transcripted in a fifth of the genome. Actually, they're even seeing function in immune responses with the section of DNA called ERVs. So how's that wrong?
They are. I completely agree. But it's incorrect but it's incorrect to assume that they're not actually retroviruses just because they have function.
Well, function would be a prediction because obviously viruses do not just randomly insert tens of thousands of, say, endogenous retroviruses, which just happen to be in exactly the right location and having exactly the right sequence to perform some valuable function. I mean, that seems like a huge leap to say that these endogenous retroviruses are evidence of universal common ancestry. I mean, how do you explain that, Kyle? Go ahead.
Well, Douglas Adams, the writer of Hitchhiker's Guide to the Galaxy, put it quite beautifully when he said that what you're saying is very similar to a hole being filled with water and then being amazed at how good of a shape that the water fits into the hole, right? So basically, what that's saying is that these sequences, these gene sequences, insert themselves into humans or other life forms, and they can end up gaining functionality or contributing to functionality, but they can also be detrimental to the host.
And I understand what you're saying. Obviously, but the thing is, these ERVs could also be some type of weakness in the system. But I think the fact that we are seeing, functionality in it, and obviously, you guys got to develop, a type of story that they could become functional. The key word is could or might. But there's also cell processes, and you would know this based on your education, that are designed to weed out harmful or viral infected genes, which is called what? Apoptosis. Is that how you say it? So after millions of years, why do we still have, according to you, so many viral infected ERVs? Because shouldn't this process have ridden them long ago? Because apparently it's not doing it at all. I mean, help me understand.
No. A good example. So ERVs are just a part of gene flow. And around like 10% of our genome is made up of viral DNA. And so it's just a common thing. We can look at another example of beneficiality through gene flow is looking at sweet potatoes. Around 8,000 years ago, Agrobacterium infested sweet potatoes and provided them a plasmid. A plasmid is a circular form of DNA that is able to pass on through conjugation. And Agrobacterium is one of the very few bacteria that is able to conjugate with a eukaryote. Usually they cannot conjugate with eukaryotes, but Agrobacterium can. It evolved the ability to do this to plants, and it would create these tumors on plants for additional food sources. But it did it to sweet potatoes, and it modified sweet potatoes to be sweet potatoes that we see today. And so it modified the traits, and it was shown to be beneficial for the sweet potato, and so it incorporated the genes from Agrobacterium into the sweet potato, and the sweet potato benefited from it, right? Well, that's what viruses can do. Not all viruses are going to just be strictly detrimental to the survival of a species.
I understand that obviously not all of them will, but with the amounts that we see in the genome that you're saying is, based on retroviruses implanting their genetic material, we should still see a lot less based on that process known as apoptosis. And the fact that we see functionality in it, it's definitely consistent. I mean, whether you have an explanation or not, whether it's a rescue device or whatever it is, I mean, it's still consistent with my model. And the debate today is which model is best. And, I think through our discussion, it's been shown that, there are limits. It's very, very hard for the evolutionists to explain even one information adding mutation. It's very hard to explain away genetic entropy, especially, in higher genomes. So at the end of the day, we'll leave it up to the viewers to watch and conclude. We've been going at it for about a half an hour. I'll leave you with the last word. Take a couple minutes, summarize yourself, your position, tell us why you believe that your model is more superior, and we'll call it a day for now. Thanks. Okay. Thanks, Kyle.
Well, to finish up, I will say that I did cover adding information, which is addition mutations and duplication mutations. Both add information. I did cover the idea of being able to change and evolve, and explaining that in order for an organism to evolve beyond its limits, we have to understand their life histories. So we wouldn't get cats the size of elephants, because the bone structures are important, and the bone density is important if you end up increasing to certain sizes, because you need to be able to survive gravity and stuff like that. And that if you actually look at the models used, which one is used in industry? And that's really the gold standard right there. Which model is used in industry? Call up any molecular biology lab, any biotechnology lab, any informatics lab, and ask them, do you look at a creationist model to do your work, or do you assume a evolutionary model to do your work? And how useful is that evolutionary model when you are actually doing your work? And I bet you that every single time they are using the evolutionary model and understanding evolution and change in species in order to do their work. And no one is using the creationist model except to sell videos to religious evangelicals.
Awesome. Well, Kyle, thanks for coming on. I think we actually, managed to have a respectful conversation. It's kind of funny on Facebook how if you read over certain conversations,
I'm sorry, you cut out a little bit. What was that?
It's funny how we can have a respectful conversation here, but it's funny when you look at like Facebook conversation, you're just reading it. It's like, wow, there seems like there's so much animosity. But it's good to be able to have a nice conversation here, Kyle, and hopefully we can do it again, brother. You have a good night, okay?
Yeah, I completely agree with you. Sometimes I can come across as kind of a bit of a dick.
Well, because I was looking at some of your conversation, I was like, wow, he's calling them morons and this and that. And all of a sudden, we're chit-chatting here. We're having a pretty, decently intellectual conversation. I think a lot of people will profit from this. And hopefully we can do it again. And I would suggest this Wednesday, Dr. Nathaniel Jensen and Dr. Mays are debating on that non sequitur show, and I think it's going to be amazing. So hopefully you tune into that as well, brother.
Could you send me the information so I can have it?
Are you talking about on this video or on the Dr. Nathaniel Jensen debate coming up?
Oh, both actually. That I was originally looking at the Dr. Jensen one.
Yeah.
Both would work too.
I think it's going to be great because so far Dr. Mays has debated Ken Hovind and Charles Jackson, but I think Jensen, especially in his book Replacing Darwin, he makes a really good case for biblical creation. And Dr. Mays, I'll admit, is a very formidable, apologist and debater. So I think it's going to be good, and I think we'll have some fun with it. So I'll send you information on that, and this video is going to upload right away, so you can go watch it. You can upload it, do what you got to do. Okay, brother?
Well, I will say one thing. Since you did bring up Kent Hovind, sure. He really does a disservice to the creationist movement because he brings up so many easily debunked and refuted points that it removes any semblance of credibility that you guys might have.
How would you feel about some of the I mean, I like Ken Hovind. I think, especially if you look at some of my debates that I've had, if I debate somebody who's not, they want to debate evolution, but they're not that knowledgeable about it. I feel like Ken Hovind's arguments, you don't even have to get too technical. They work. When I'm debating someone like you, when I'm debating, someone like, I actually enjoy the more technical, intellectually, type discussions. And then, you just kind of go from there. But who would you say would you say it's Dr. Jensen would be our top apologist right now? Or what do you think about that?
Actually, I am a huge fan of Todd Wood.
Okay, yeah. He was in
his
Genesis History, right?
Well, I don't know too much about Jensen at the moment. I'd have to refresh my memory. But Todd Wood is I have a lot of respect for the man. He's very honest, and if you look on his website, which is like a blog, he's an Answers in Genesis speaker, right? And he admits on his blog that there is evidence for evolution, a lot of evidence for evolution. It is scientific, and he admits that it is not a failure in any sense, and it's used often, and it works as a model. But he says that he doesn't accept it in its most logical conclusions of like universal common ancestry. He thinks that there's another explanation, but at least he admits that evolution does have evidence, and that's why I respect him, because he can understand why we're saying what we're saying.
No, and I think I've seen that blog. I do like him. I do respect him. But yeah, let's do this again sometime. It's going to be uploaded right away. My wife's calling for me. So, hey, you have a good night. Get some sleep and enjoy. I know you start a new job tomorrow. So have fun, man. Best of luck.
Thanks. Have a good one.
Okay, man. Take it easy.